This is Chapter 11, Part B, on nervous system and nervous tissue. So, like all cells, neurons have what's called a resting membrane potential. But unlike other cells, neurons are able to rapidly change this resting membrane potential and generate what are called action potentials or electrical signals. So, this just means neurons are highly excitable cells. So looking at some basic principles of electricity. So we all know opposite charges are attracted to one another. So it requires energy to keep those opposite charges from naturally wanting to join together, to keep those charges separated. So think if you're holding two magnets together and they naturally would want to connect and be drawn to one another, but you apply energy with your muscles to physically keep them separated. So this energy is finally liberated when the charges are allowed to move toward one another. So when these opposite charges remain separated, it's considered to have a potential energy. So it has the potential to release energy, it just hasn't done it yet because those charges are still separated. So as we know from the muscle chapter that the resting membrane potential is more negative than the outside. So when we look at depolarization, it's going to be a flipping or reversing of that charge difference. So a few electricity definitions. When we talk about the voltage of the membrane, it's just a measure of that potential energy that's separated or generated by those separated charges. So it's measured in either volts or millivolts, and sometimes referred to as the potential difference or just the potential. So when we talk about the membrane potential, we're just talking about the membrane voltage. So that charge difference across the membrane is what results in our potential or our voltage. So generally the greater the difference between the 2 points, so the steeper that gradient, the steeper the hill between those two charges, you would have a higher voltage. So this is showing some charge differences. So at a resting state, it's more negative inside the cell, right? And it's more positive outside the cell. So this is showing the differences in these ion concentration gradients. So there's more outside than inside. So these positively charged sodiums would be drawn toward the inside of the cell where it's more negative. Right? And these negatively charged chlorines or chlorides on the outside would be more attracted to the positively charged potassium ions in the membrane. So when we talk about current, what we're referring to is just a flow of electrical charge or a flow of ions between the 2 points. So this current can be used to do work, and it's going to depend on that voltage and resistance. So again, the current would be the influx of the sodium ions when we looked at muscle contraction. So once we get that signal from the nervous system and we open those sodium channels, then we get a current of that positively charged sodium moving inside the cell. So resistance is just anything that's going to hinder that flow of ions or electrical charge. So when we look at resistance, we have things that are insulators. So this would be something with a higher electrical resistance. So it's going to kind of slow that current or that flow of electricity. Anything that is a conductor is going to have less resistance. So it's going to help conduct and transmit that electricity or that signal much more rapidly. So Ohm's law shows us the relationship between this voltage and current and resistance. So it basically states that current is directly proportional to the voltage, right? So the flow of the ions or the electricity, sometimes measured in amperes or amps, is going to be proportional to the voltage. So this means if we increase the voltage, then we're also going to increase the current or the flow. So think of this as the voltage that's pushing our current. So the stronger we're pushing that current, the faster it's going to go. So current is then inversely proportional to resistance. So if resistance goes up, then our current is going to go down. So think of resistance as kind of tightening the noose or narrowing this channel, right? So it's going to slow down our flow. So the greater the resistance, the smaller the current, right? So the less the flow. So membrane channels play a large role in this voltage change across the membrane. So they're going to serve as our selective membrane ion channels. So two main types. So the first ones are leakage channels, sometimes referred to as non-gated. So these are basically always open, always leaky. Pretty much anything that will fit through these channels is able to pass through. A gated channel is going to involve the protein actually changing its physical shape to either open or close, right? So it has kind of an actual gate. So we have to have a particular key to unlock these gates on these channels. So there's three types of gates that we can find. So the first ones are chemically gated or ligand gated. So this just means that they're only going to open if a specific chemical or neurotransmitter binds to that protein, to that channel. So going back again to our example with muscle contraction. So it was the acetylcholine, right, that binds to those sodium channels and then allowing them to open and sodium ions to then move inside the cell. A voltage-gated channel is going to open and close in response to changes in the membrane potential or the voltage of the membrane. So at the resting potential, negative 70 millivolts, they'll be closed. Right? So we're at rest, we don't need to open the channels. But once we reach that threshold, so again, going back to muscle contraction, so once we reach that tipping point, the voltage changes to negative 50, so it's gained about 20 millivolts. So now that's going to trigger these channels to open, and that's when we have kind of that exponential rise of the action potential. So now we have more sodium ions influx inside of the cell. Mechanically gated channels are going to open and close in response to some type of mechanical stimulus or a physical deformation of the receptor. So these are more like your sensory receptors, maybe some of your pressure and touch receptors. So whenever these gated channels finally open, the ions are going to be allowed to diffuse and enter the cell quickly. So they're still going to travel from high concentration to a low concentration. So that standard diffusion is still going to take place. So we're going to go from high concentration to the top of the hill and then flow downhill, right, where the lower concentration is. So the electrical gradients are going to move toward their opposite charges. So in the resting potential, right, we said it was negative inside. So once the ions start to diffuse, these positively charged ions like sodium, when they enter the cell, they're also carrying that positive charge with them. So now the membrane potential or inside of the membrane is going to become less negative or more positive. So when we look at cells and neurons, we look at the electrochemical gradient. So we're basically combining the electricity and the ions and the chemical. So the ion flow is going to create our electrical current and ultimately change the voltage across the membrane. So again, the resting membrane potential is about negative 70 millivolts. Right? So this just means that the inside of the cell is negatively charged compared to the outside. Right? So this is said to be a polarized membrane. So instead of having like a north pole and a south pole, we have a positive pole and a negative pole. So this membrane potential is generated by the differences in the ionic compositions inside and outside the cell. So there's just naturally more sodium in the cell exterior, so in the fluid of the cell, than there is inside the cell. Right? So same thing with potassium. We have more potassium generally inside than we do outside. It's also generated by differences in the plasma membrane permeability, right? So going back to those membrane channels. So if we open some channels and allow this sodium to come in, then that could affect our membrane potential or membrane voltage. So like we said, the extracellular fluid, the ECF, has a higher concentration of sodium than the intracellular fluid. Um, so this is balanced mainly by the negatively charged chlorine ions. So ICF also has a higher concentration of positively charged potassium, but it's going to be kind of balanced out and zeroed out by the negatively charged proteins in the cell. So we have lots of proteins, plasma proteins, and things in the cell that carry negative charge to balance out this positive potassium. Right, so ultimately, potassium is the most important in maintaining our resting membrane potential. So this is just showing the different concentrations of sodium and potassium on either side of the membrane. So outside, we have more positively charged sodium than we do inside. Right? So the sodium-potassium pump is going to help to maintain these natural concentrations and gradients across the membrane. So our membrane potential can change if our concentration of ions change. So maybe we have more sodium come into the cell and bring its positive charge with it. Membrane potential also changes when the membrane permeability to ions change. So again, think back to muscle contraction. So we had our chemically gated channels that open for sodium in response to acetylcholine, and then we reach that threshold tipping point to open up our voltage gated channels. Right? So that's going to increase our permeability to sodium. So now there's more sodium channels open, there's more sodium allowed to come in. So we use these changes in membrane potential to send signals, right, to send and receive information throughout the body. So there's two main types of electrical signals we can generate using this resting membrane potential. So the first ones are graded potentials that we mentioned briefly in part A. So these are short distance signals. So basically from the dendrite to the cell body, and that's it. Right? So they don't have as much voltage behind them. They don't have as fast of a current because they don't have to travel as far. Whereas your action potentials, these are your long distance signals. Right? So again, thinking back to muscle contraction, so the signal can go all the way from your brain, your central nervous system, all the way down to the muscles in your pinky toe. So these are action potentials traveling long distances down the length of the axon. So some terms describing our membrane potential are going to be relative to that resting potential. So sometimes when we say um, that it's become positive, even though it's still negative 55, because relative to negative 70, where we started at, it is a more positive voltage. So when we talk about a depolarization, then we have essentially decreased the membrane potential, so it has become less negative. So we've essentially kind of reversed and flipped that membrane potential, so now it's less negative inside than outside. A hyperpolarization is when we increase the membrane potential further away from zero. So we actually go a little bit beyond that resting potential and make it the inside of the membrane more negative than it would be at rest. Right? But this is kind of like a refractory period to reduce the probability of producing another impulse. So we want to have one complete cycle of action potential generation and propagation before we stimulate the neuron again and try to do another round of action potentials. So again, once those gated ion channels open, the depolarization is going to spread or propagate from one area of the membrane to the next. So kind of like a ripple effect. Um, so The direction of the impulse is going to go from the cell body toward the axon terminals. So at the beginning of the axon, we have our voltage gated channels, right? So this first section of the axon has been depolarized, so now it's less negative inside. So we have our positive charge inside. So this is going to trigger these adjacent voltage gated channels to start to open. So they'll allow sodium in down here. So the depolarization at one segment is going to trigger opening of channels in the next segment. Right, so we have kind of a snowball effect. So it's just going to spread and propagate all the way to the end of the axon. So action potentials are said to spread kind of like a wave of depolarization. So once the depolarization has finished and moved on from a particular segment, then the previous segment will start to repolarize to return back to that resting state. Like we said, action potentials are the principal way of long distance communication in the nervous system. So we've already talked about action potentials in muscle cells. So the process is essentially the same in neurons. So it includes that brief reversal of the membrane potential, meaning that we're flipping the membrane potential to be less negative inside. So we can have a change of up to 100 millivolts. We can go from negative 70 all the way to positive 30 when we depolarize and change this membrane potential. So action potentials are good for long distance communication because they do not decay over long distances like our short distance graded potentials do. So when we talk about a nerve impulse, what we're referring to is this spike of action potential, this depolarization of the membrane. So it's going to involve opening of those specific voltage gated channels. Right. So again, we have maybe our chemically gated channels opening here. So we start to decrease the voltage. So once we reach this threshold, this tipping point, this is then going to trigger opening of our voltage gated channel. So this is when the spike really hits its peak, and we have kind of that snowball effect of more sodium influx inside the cell. So there's four main steps to generating an action potential. So step number one, at the resting state, all of the channels are closed, right? So we're not stimulating the cell, everything's closed, everything's at rest. So it's that negative 70 resting state. So this is going to help maintain our membrane potential. So we don't want to open the channels until we're ready for an action potential. So step two would be depolarization. So now we've gotten the signal, we can open our sodium channels. So once these channels open, sodium will rush into the cell. And again, because sodium carries a positive charge, it's going to kind of make the inside of this membrane less negative. Right? It's going to carry that positive charge with it. Right? So as a result, the ICF becomes less negative. Right? So once we hit that threshold, about negative 50 millivolts, we have this positive feedback kick in. So again, kind of a snowball effect. So now we're going to open the rest of our sodium channels. Right, and get this large action potential spike shown on the graph. So the membrane polarity jumps to about positive 30. So we have a very, very rapid change of about 100 millivolts from negative 70 all the way to positive 30. So then step three, after we've generated the action potential, right, it's going to spread down the axon. Our initial segment of the membrane will begin the process of repolarization. So this is essentially resetting everything back to that resting state. So we're going to start to close our sodium channel. So we stop the influx of sodium and we open up some potassium channels to let some of that positive charge exit the inside of the cell. Right, so repolarization, we're returning to the resting state. You see this decline now go on the graph. So then finally, step 4 is hyperpolarization. So some of the potassium channels remain open. So we have a bit of excess potassium leave the cell. So this is going to cause the membrane to become a bit more negative than it would at rest. We dip a little bit below that resting potential. Um, so again, you can think of this kind of like a refractory period. So it's just ensuring that we have one complete action potential cycle and process, and we've completely reset before we start another action potential generation cycle. Right, so just showing all the 4 steps together. So step 1, resting state, everything's closed, nothing's happening. Right, depolarization, we open up the sodium channels. Those positively charged sodiums come in and flip our membrane potential. Right, so we get our depolarization and our action potential peak. So then repolarization, we start the process of resetting back to that resting state by closing the sodium channels. We stop the influx of sodium and we pump out some of that positive charge with the potassium. And then finally, step 4 was hyperpolarization. So some of those potassium channels remain open and we lose a little bit of excess potassium that lowers our membrane potential below the resting state. So propagation is how the action potential is going to be transmitted from where it's initially originated, where we have our initial depolarization, all the way down the length of the axons until we get to those axon terminals. So again, sodium influx through the voltage gates in one membrane area is going to trigger opening of sodium channels in a neighboring or adjacent area of the membrane. So this leads to depolarization of that area, which in turn causes depolarization of the next area. So again, like a snowball effect. So depolarization here is going to trigger opening of voltage gated channels here. All right, so same thing here. When we have depolarization here, then we're going to stimulate the voltage channels over here to open. So this depolarization, this action potential, is going to persist down the length of the axon. Right. So again, the refractory period is that period after the action potential spikes all the way to the end of hyperpolarization. Right. So this is just when our channels are closed, so the neuron cannot respond or trigger another action potential until we have completed our entire cycle. So looking at conduction velocity, we said that the action potentials only occur in the axons, so not other cell areas. So the conduction speed or velocity is going to vary among different types of axons. But the rate of propagation or the speed at which these impulses travel are going to depend on 2 main factors. So the axon diameter. So your larger or your thicker fibers, thicker nerves, are going to have less resistance. So therefore they would have a faster impulse conduction. So kind of like a highway. So the more lanes you have open on a highway, the faster and smoother traffic can flow, as opposed to a very narrow road, right, where traffic would be more backlogged and slow down a bit. Also, the degree of myelination. So that myelin that helps to insulate and conduct those impulses is going to, how much myelin a neuron has is going to have an effect on how fast that neuron can conduct these impulses. So there's 2 types of conduction depending on the presence or absence of myelin. So we'll look at those in just a minute, but those are called continuous conduction or a saltatory conduction. So in continuous conduction, it's a slower conduction that's going to occur in your non-myelinated axons. So continuous conduction essentially means that we have to undergo this process of depolarization at every instance along the axon membrane. Right, so we'll have to open channels here and depolarize, open channels here and depolarize, and so on and so forth until we reach the end of the axon. So this process just takes a little bit more time. Whereas your saltatory conduction, sometimes referred to as the jumping conduction, is going to occur in axons that are covered with the myelin sheath, with these Schwann cells. So this is actually about 30 times faster than your non-myelinated fibers. So we said that myelin helps to insulate the charge, prevent leakage of the charge. So the voltage-gated sodium channels are located at these sheath gaps, right, at the nodes of NVA. So the electrical signal apparently can jump from gap to gap. So instead of having to depolarize along every square micrometer of the axon, right, it can kind of hop between the sheath gaps. Right, so it can travel much more quickly and efficiently. So just another figure showing the difference between your continuous conduction and saltatory conduction. So in a non-myelinated axon, so a bare, naked axon, the voltage-gated channels have to open at every point along the axon. So that way we don't lose the power or the intensity of our charge. Whereas in your myelinated axons, it can kind of take a shortcut and skip ahead certain sections and jump rapidly between these gaps. So multiple sclerosis is a well-known homeostatic imbalance of the nervous system that affects the myelin sheath. The multiple sclerosis is an autoimmune disease where the individual's immune system cells actively attack and destroy the myelin sheaths in the central nervous system. So it turns that myelin into hardened kind of scar tissue lesions called scleroses. So this ultimately causes that impulse conduction to slow or eventually cease altogether. So some symptoms of multiple sclerosis include visual disturbances, weakness, loss of muscular control, speech disturbances, incontinence, and generally treated with drugs that modify immune system activity. So sometimes things to help kind of dampen the immune system so it's not so active that it's actively attacking those nerve cells. So multiple sclerosis is something that I know a lot about. Unfortunately, my mother has MS for about 15 years now. But something interesting I've learned throughout this, one of the medications she used to take called Rebif is just a beta interferon. So this is a substance that's produced naturally by the immune system. But in order to harvest this beta interferon for medication purposes, it's produced by recombinant DNA technology. So we have genetically engineered hamster ovary cells to make this human beta interferon gene and product. So looking back at conduction velocity, so nerve fibers again are classified based on their diameter, how much myelin they have, and how fast they can conduct these impulses. So they're going to fall in three major categories. So your group A fibers are the largest diameters, they have the most myelin, and they have the fastest transmission speeds, right? So up to 120 meters per second impulse conduction. So these are found mainly in your somatic, sensory, and motor fibers in your skin, like the skeletal muscles and the joints. Your group B fibers kind of are intermediate diameter. They're not as thick. They have light myelination, not as much as your group A. And they conduct impulses around 15 meters per second or so. So group C are the smallest diameter and are unmyelinated. So in this figure, the myelin is pictured in yellow. So you see the group C fibers are basically just raw axons. They have no myelination whatsoever. So you can also see that these are going to transmit at the lowest speeds as well. So your group B and C, because they conduct more slowly, are typically found in your autonomic, visceral, motor, and sensory fibers that serve the visceral organs. So things like your digestive system that doesn't really rely on that very rapid speed conduction for things like reflexes. So another type of homeostatic imbalance could be caused by an impaired action potential impulse propagation. Um, so could be due to a number of chemical or physical factors. So maybe a local anesthetic like Novocaine, lidocaine works by blocking those voltage-gated sodium channels. So if we block those channels, they're never able to open, so therefore we would never get an action potential generated in the first place. Um, Cold temperatures or continuous pressure that interrupt the blood circulation to those neurons can also cause an impaired impulse conduction. So, like when your hands get really cold and your fingers get kind of numb, or if you sit on your foot, sit the wrong way for too long and your foot goes to sleep, basically due to pressing on the nerves and cutting off the blood flow to the nerves. So they're not really able to generate those action potentials and work as properly.